Welcome to the online class offered by the Department of Botany, Marthoma College, Chungapuram. Today we will discuss more details on the topic genetic engineering. First, we'll see what are the different tools used in genetic engineering. So first we'll discuss what genetic engineering is. Genetic engineering is also known as rDNA technology, that is recombinant DNA technology. It is, in simple words, the changing of genes by using in vitro processes. So a gene is modified. So in this sense, a DNA is modified to perform a specific function. So it will be improved to have a specific character and to perform specific functions. A gene of known function can be transformed from its normal location into a cell through a suitable vector. So here, from one organism, a particular gene can be transferred to another organism. A desirable change can be made by this transfer. So this gene which is transferred from the other organism or the other cell is later replicated in the host genome, and this presence of the new gene can thus be confirmed by biochemical analysis. So after this process, the new gene becomes the part of the host genome. Derivation of procedures for the reintroduction of foreign DNA material into a host have led to the evolution of a new technology, and that is recombinant DNA technology, what we call genetic engineering. So here there is the first step of identification of desired gene and isolation of this desired gene, incorporating it into the host genome, and we will also analyze how this gene is expressed in the host genome. Gene manipulation is the deliberate and directional manipulation. So here the genetic material or genetic makeup of the organism or of a single cell is changed in a predetermined way for producing a genetically altered cell or an organism. So finally what we get is a genetically modified organism, what is commonly known as a GMO. Many genetically modified organisms are known. So what we commonly call the BT crops like BT tomato, BT brinjal, BT cotton, etc. There are also genetically modified microorganisms like bacteria which are able to perform specific desirable functions like producing an industrially important chemical or producing therapeutically important compounds, etc. So manipulation of genes in a desired manner under controlled conditions for genetic modification of living systems. So here the living system is being modified by the change in genetic makeup. So the genetic makeup gives a change in the RNA, and finally this RNA gives a change or an altered form in the protein makeup, and thus a changed phenotype is expressed by the organism. It is the most fundamental aspect of biotechnology, that is the manipulation of genes in a desired fashion or in a desired direction. So it involves the generation of a recombinant DNA by physical combination of previously and evolutionarily unrelated DNA sequences from different sources. This point is elaborated and the whole process is described. Here we can see that there are two important features to be taken care of, that is DNA sequences which are recombined are from two different sources. That is, they can be between unrelated species or different types of organisms. This means that a bacterial genome can be incorporated into a plant genome, or a plant genome can be added to an animal genome. So, thus, unrelated species can have their characters crossed with each other, and this recombinant DNA is expressed in the genetically modified host organism. The different goals of recombinant DNA technology, it is of various aspects, like preparing a gene library. So we have a collection of genes from which we can take or we can receive various desired genes for further research and uses. Then we can identify, isolate and amplify desired genes. So for a research purpose, if we require a particular gene, we can identify it from the collection and we can isolate and amplify it. This also is a goal of recombinant DNA technology. Another one is understanding the expression of specific genes and its regulation. How specific genes or certain genes function and how it is expressed and how this gene is regulated in the organism. How it is controlled, it is stopped or it is allowed to function. So these are all understood clearly by the various steps carried out in rDNA technology. So from now on, we'll be mentioning rDNA technology instead of the whole big name recombinant DNA technology. We can also carry out various alterations or modifications in the expression of selected genes. So certain genes can be controlled, the level of expression can be increased or decreased, it can be altered to specific conditions. So such changes can also be made by the rDNA technology. We can also alter the pattern of gene expression, how it is expressed in certain cells or certain organisms. So all these are the different goals of recombinant DNA technology. And coming to applications of recombinant DNA technology, the molecular basis and resolution of specific human disease genes and the treatment of hereditary diseases. So this is a very commonly applied field of or use of genetically modified organisms and cells. So the various bases on which different diseases are caused to human beings and other animals are understood clearly, and also we can give treatments to certain hereditary diseases. So diseases caused by mutations or defects in genes can be treated properly and effectively these days by this rDNA technology. We can also carry out large-scale production of therapeutic human proteins like insulin, immunoplasmin, somatotropin, etc. Also, vaccines are produced in large amounts. So we use certain plants, products of plants, or even microorganisms for producing vaccines in large scale. So all these are carried out with the help of rDNA technology. Modification of useful plants and microbes is also next application of rDNA technology. So these animals and plants or microbes are used to produce crops or products with improved quality and quantity. In the image, we can see how, in simple ways, the rDNA technology is explained. So, the plasmid gene from a bacterium is taken, it is cut using enzymes. Another desirable gene, here human insulin gene is added to this plasmid, and it is again taken into the bacterium where this modified gene is being replicated or amplified. So multiple copies of this gene is produced, and the insulin is produced in large scale. After this, from the bacteria, insulin is extracted, it is purified and ready for. Next, we'll see different processes involved in rDNA technology. The first and foremost step is the identification of source of desired gene. Here, we have to identify the specific character which we require and thus the source of this character, from which organism we can get this desired gene. After identification of the source, we isolate the desired gene or the DNA. In this step, we can carry out the process in two different ways. We can either isolate it from the parent organism where this desired gene is present, or we can artificially synthesize it comparing the genome of the parental cell or the source cell. By these two methods, we can obtain the desired gene or the DNA. In the third step, the in vitro incorporation of isolated gene or DNA segment into small self-duplicating recipient DNA is done. Here, many chromosomes or vectors are being used. This combination of the desired gene with the mini chromosome is known as recombinant DNA, and this process is recombinant DNA technology. After incorporating it with the chromosomal DNA, it is transferred to the host cell and allowed to amplify. So this step is gene amplification or gene cloning. Here, the recombinant DNA is maintained within the host cell and it is allowed to multiply in the host cell. In the next step, the cloned gene is introduced into the recipient or the target cell. So here comes the final recipient of the recombinant DNA. So the effect or the final product is obtained from this final recipient. The desired gene is thus transferred to the host cell by suitable vector. So those vectors which are used to transfer the character or the gene is known as transfer vector, and those vectors which are used for amplification of genes are known as amplifying or cloning vectors. There are two different types of vectors, transfer vectors and clone vectors. rDNA technology permits gene transfer between different species or genera which do not interbreed. This is the major plus point of rDNA technology because it allows the gene combination between normally unrelated species, genera, or even organisms. These are two images which show transformation or recombinant DNA technology in plants. Here, plants gene is modified using the vector Agrobacterium tumefaciens. Here, the tumor-inducing plasmid is connected or combined with the suitable DNA, and it is added back to the agrobacterium, which infects the plant and inserts the specific DNA into the plant. So here, this agrobacterium acts as a vector, and we have obtained the desired gene from some source, or we either artificially synthesize it. Then we combine these plasmid DNA with the foreign gene and allow it to grow inside the agrobacterium. And this agrobacterium infects the particular plant and adds its genome into the plant cell and the plant cell or the plant is converted into a genetically modified plant. The second picture also describes genetic engineering process in 3 steps. Single gene is isolated and modified. So the cells with desired trait is selected, and the DNA required or responsible for it is selected and isolated. So isolate the target gene. Many copies of the gene are inserted into the plant cells. So that is done by this Agrobacterium in plants normally, and then it is allowed to grow in the plants. So from this mature plants, seeds are produced which carry this modified gene. These two pictures describe the various gene transformation or rDNA technology carried out or followed in plants. Here, the purposeful alteration of phenotype of an organism in most desirable manner is carried out. So normally it is done for scientific, medical, agricultural, industrial or commercial purposes. Here we permanently add the foreign genes into the target cell. In this case, we have to consider the various needs which is commonly either for research purposes or for treating some disease or for improving some crops as in agriculture fields, can be animals, plants, for producing specific desired compounds in large scale that is seen in industrial purposes, and for commercial purposes for producing modified organisms. It can be plants, organisms, etc. Two basic discoveries which was done in the field of molecular biology and biotechnology have helped in many ways to carry out these processes. So the modern rDNA technology is very thankful for the scientists who have discovered these two major discoveries. The first one is the molecular organization and functional features of DNA, which is the discovery of the structure and characteristic features of DNA. The second one is the discovery of enzymes which are able to cleave, elongate, ligate and modify DNA. These two discoveries paved way for the modern rDNA technologies. Natural gene transfer is also existing. So normally this process is taking place during sexual reproduction in almost all organisms, in bacterial transduction and transformation techniques, and so on. But this has a limitation. That is, this process is carried out between related species or same species, two individuals of same species. Here we cannot carry out intergeneric breeding or between two organisms or even between two families. In this condition, rDNA technology is an advantage to this limitation. So we can say that rDNA technology is an efficient way to transfer desired gene to target cells and modify them in a desired fashion. There are various requirements for these rDNA technology. We have seen the different steps required for the process. Now we'll see which are the different requirements needed for the process. The first and foremost requirement is the donor DNA which has got the desired gene. So we have said some gene which is having the desired character is to be identified or understood. It is identified from a particular source and it has to be isolated. So the parental or the host genome or the donor DNA, which is the desired DNA, is to be understood properly. Second one is a vector DNA or a carrier DNA which carries this desired gene to the target's gene for gene amplification. So the vector DNA is also important. which carries out the amplification and the cloning process. The third requirement is the host cell, which is to be modified. Some cell or organism has to be modified. That is the third requirement. It is the host cell which accepts the recombinant DNA. The last requirement is a major requirement, that is the enzymes which are needed for various steps. So only in the presence of enzymes the desired gene can be cut, it can be added to the vector DNA, it can be added back to the vector cell and amplified and taken to the desired gene or the target gene. Here all the four requirements are major requirements itself, and we need all these to carry out the different processes. Along with these, there are certain characters or conditions which are required to carry out successful rDNA technique. With this, we conclude the general introduction about the various processes and tools required for genetic engineering. In later classes, we'll discuss what are the different tools in detail, what are the enzymes, which are the vector cells, which are the host cells, and how we can obtain the desired gene from a particular source. Thank you.